Okay, this is part 2 of lecture 7. So we'll go into cellular respiration. So cellular respiration has 3 stages, as mentioned in the intro. There is glycolysis, pyruvate oxidation and the citric acid cycle, and then oxidative phosphorylation. So I'll go through one by one, and I put headers that are numbered to kind of help show, okay, this stage 1, glycolysis. So glycolysis, as you see in this diagram, happens outside of the mitochondria, and you make some ATP here. So glycolysis happens in the cytoplasm. It can happen, like anywhere in the cell outside of the mitochondria. So glycolysis happens in the cytoplasm and has 2 major phases. There is one part where you put in some energy. So it's the energy investment phase. So 2 ATP are used to be able to split glucose into 2 3-carbon sugar molecules. And then the next phase is the energy payoff phase. So in this phase, 4 ATP are made. 2 molecules of NAD+ are reduced to NADH. And the small sugars are oxidized to form 2 molecules of pyruvate and 2 molecules of water. So you invest 2 ATP, and then you make a total of 4 ATP, so that means a net of 2 ATP are produced. And this is made by substrate level phosphorylation, which means there's an enzyme that transfers a phosphate group from something onto ATP, or ADP to make ATP. So here is like an overview of the phases. So this is the energy investment phase. And so you feed in 1 glucose molecule. You use up 2 ATP at this point so that you can use that energy to split the glucose molecule. So at this point you used up 2 ATP. But in the energy payoff phase, you make 4 ATP. And then you also happen to make 2 NADH and 2 molecules of pyruvate. And then there's water produced as like a byproduct. And then there are also, because the electrons are transferred, when the electrons are transferred, 2 protons are also transferred, but they kind of just get put into the cytoplasm. So in total, if we look at the inputs and outputs, we took glucose and chopped it into 2 molecules of pyruvate and some water. We're looking at the ATP, so there's 4 ATP formed minus the 2 used, so you make a total of 2 ATP at this point. And then you go from 2 NAD pluses to 2 NADHs. And then there's some protons in solution as well. So those are like the overviews, what you put in, what you take out. So at this point, all of the carbon that's originally present in glucose is now rearranged and is present in those 2 molecules of pyruvate. And at this point, glycolysis doesn't release any carbon dioxide. The carbon is still in the pyruvate molecules. And glycolysis can happen whether or not oxygen is present. And so glycolysis is actually like a really, really, really like an ancient pathway that almost all cells use glycolysis. So glucose gets broken up, and so all cells need some form of energy, and most of them are going to get it from either glycolysis or then continuing on the rest of the cycle. So just to show you the like details of how you get from glucose to pyruvate. So there's a bunch of different steps, and so each one of these steps is catalyzed by an enzyme. So in this first step, there's an enzyme called hexokinase. So remember how I said every enzyme ends in ase, or almost every enzyme. You could see that it's true for all of these guys. So they end in ase. So hexokinase removes a phosphate from ATP and then slaps it onto glucose. And so right now, this is glucose 6-phosphate. It's glucose and it has a phosphate right there on the 6th carbon. Then there's another enzyme, phosphoglucoisomerase. And so what this one does is just kind of like rearranges stuff. So now it's not like a little hexagon, now it's a pentagon. So it kind of shifted one of the carbons over here. So now this is considered fructose 6-phosphate because it's a different shape. In chemistry, every single change matters. That's a whole nother name for this molecule. So now this is fructose 6-phosphate. Again, this enzyme, phosphofructokinase, uses a phosphate group and adds it onto this other part of the molecule. So now this is fructose 1,6-biphosphate because there's a phosphate on here and at 2 different positions. So the biphosphate means that it's 2 different positions, and then this, it tells you which carbons are on. So if you are a bio major, at some point you're going to have to take general chemistry and organic chemistry, and it's going to be a lot of this, unfortunately. But it is really all relevant. Chemistry really plays a lot into biology. So, sorry. Then there's a 4th enzyme called aldolase that kind of chops this guy into 2. So now you have 2 molecules of what are called G3P, which stands for glyceraldehyde 3-phosphate. So glycerol, and then there's an alcohol, and then a phosphate. And then so one of those is G3P, and then the other one is DHAP, dihydroxyacetone phosphate. And then those are altered by isomerase. And so, yeah, each point is catalyzed by an enzyme, and at the very end, you have pyruvate, 2 molecules of pyruvate. So this was the energy investment phase. You could see where the ATP was put in. And then this is the energy payoff phase. This is where 2 ATP are made here, 2 more here, and so you make a net of 2 ATP total throughout glycolysis. And then this is also where you see the NADH get made. So this is the big picture summary of glycolysis. You take a glucose, and they get altered in a series of steps into 2 molecules of pyruvate. You have a net of 2 ATP, and then 2 NADH. So phase 2 of cellular respiration is kind of 2 phases working together. So there's pyruvate oxidation and then the citric acid cycle. These happen inside of the mitochondria. And then you make some ATP here. I was going to change this header. So pyruvate oxidation is what we're going to start with. So most of the energy in glucose is still stored in the pyruvate molecules that are made by glycolysis. And in eukaryotic cells, if oxygen is present, then pyruvate enters the mitochondria and then can complete the glucose oxidation. In prokaryotes, because they don't have membrane-bound organelles like mitochondria, this will happen just in the cytosol for prokaryotes that are aerobic and can do these processes and use that oxygen. So yeah, inside eukaryotic cells, it's going to happen inside mitochondria, and then in prokaryotes, it's in the cytosol. So pyruvate oxidation. Pyruvate is converted to this molecule called acetyl coenzyme A, or acetyl-CoA, before it can then enter the citric acid cycle. So this happens by this enzyme called pyruvate dehydrogenase. Pyruvate dehydrogenase catalyzes 3 reactions. So it's that oxidation of pyruvate's carboxyl group. This releases the first carbon dioxide molecule of cellular respiration. So I'm pretty sure I mentioned that as a byproduct of cellular respiration, molecules of carbon dioxide get made, and that's why we have to breathe out carbon dioxide, because all of our cells are doing all these processes and building up carbon dioxide, and it can be toxic to cells, so they have to be released. So this is one of those points at which carbon dioxide is made. Another reaction that is catalyzed by pyruvate dehydrogenase is the reduction of NAD+ to NADH. So remember, oxidation is when something loses electrons, and then reduction is when something gains electrons. So it's reduced because it was more positively charged, and now its positive charge is reduced, and now it's neutral. So now reduction of NAD+ to NADH. And then there's a combination of the remaining 2 carbon pieces with coenzyme A, and this is what forms acetyl-CoA. So it's like the carbons that were all present in that glucose molecule are getting rearranged now at this point. So here you see the molecule of pyruvate on the left. There's 3 carbons, and remember there's 2 molecules of pyruvate. So 3 carbons. This is our carboxyl group. So pyruvate has to get taken into the mitochondria, and this will happen with a transport protein. Then once it's inside the mitochondria, pyruvate dehydrogenase will take off this carboxyl group, and now this is carbon dioxide. And then with the remaining ones, makes acetyl-CoA. And then it also happens to do the reduction of NADH. So now this molecule acetyl-CoA can go into the citric acid cycle. So the citric acid cycle is also called the Krebs cycle, in case you see any other like YouTube videos talking about the Krebs cycle. It's referring to the citric acid cycle. And I believe the Labster assignment calls it the Krebs cycle, but yeah, the same cycle. So the citric acid cycle oxidizes the organic fuel derived from pyruvate and ends up making 1 ATP, 1 NADH, and 1 FADH2 per turn of the citric acid cycle. I'll show the cycle in the next slides. So it's a cycle. And I'll show you on the next slide. So in doing this, it makes ATP, NADH, and FADH2, which is also an electron carrier. And so another 2 carbon dioxide molecules are also produced as a waste product. And this happens like 1 cycle per pyruvate. So because 2 pyruvate molecules are made per glucose molecule, the cycle runs twice. Anytime that you break down 1 molecule of glucose, you have to run the cycle twice, because that makes 2 molecules of pyruvate. So here is a figure showing this is pyruvate, the pyruvate oxidation, making acetyl-CoA, And so now acetyl-CoA feeds into the citric acid cycle. So acetyl-CoA goes through a series of reactions and intermediates, and in doing so makes 2 molecules of carbon dioxide, 2 molecules of NADH, some ATP, and FADH2. And again, since there's 2 molecules, it goes through twice. So it makes twice that much total per glucose molecule. And then this is really a cycle. So there's all these intermediates. It's not just like one reaction that, like here, where it goes pyruvate to acetyl-CoA. This whole cycle ends up feeding into itself. So whatever is made at the end is what's used to start the next round of the citric acid cycle. So again, you'll see what I mean in now the next slide. So the cycle has 8 steps, and each one is catalyzed by a specific enzyme. So the first reaction that happens, the first step is that the acetyl group of acetyl-CoA joins the cycle by combining with this other molecule called oxaloacetate. And then when they combine, it forms this molecule called citrate. And then the next 7 steps continue decomposing the citrate back to oxaloacetate, making the whole process a cycle. And then like the molecules that are made in the meantime, NADH and FADH2, those are going to carry electrons to the electron transport chain. So remember when I talked about redox reactions and then electron carriers, this is where that's coming into play. These molecules are kind of just holding on to electrons so that they could kind of play hot potato with them, and then carry this reaction on safely and not in one huge explosive reaction of transferring electrons directly to oxygen. So here's a figure with some more details, a closer look at the citric acid cycle. So this is acetyl-CoA. And then each one of these is catalyzed by a different enzyme. So acetyl-CoA combines with oxaloacetate and makes citrate. And then there's the next reaction that makes isocitrate, and then the next reaction that makes alpha ketoglutarate. The next one succinyl-CoA, the next one succinate, the next one fumarate, the next one malate. And then at the end, you remake oxaloacetate, and then that's going to feed into the next round. So again, because you have 2 molecules of acetyl-CoA, you're going to run this twice. So this is a big picture summary of like the inputs and outputs. So when you feed in acetyl-CoA, you have 2 molecules of that. Then what you get out is ATP, 2 molecules of ATP, 8 molecules of NADH, because again, you're running it twice, 2 molecules of FADH2, and then 2 molecules of carbon dioxide, which is a byproduct. So then stage 3 is oxidative phosphorylation. It's a mouthful. And so this is oxidative, and then you're going to phosphorylate something. So oxidative phosphorylation actually has 2 parts, 2 stages. There is the electron transport chain and chemiosmosis. So together that makes the whole process of oxidative phosphorylation. So just as a preview, there's a bunch of proteins here in this membrane. And so this part, the electron transport chain, even though we're separating them, like it works really closely because chemiosmosis is right after. Like this drives this to happen. So oxidative phosphorylation has 2 stages that have to happen together. So those molecules of NADH and FADH2 that are made during glycolysis and the citric acid cycle have most of the energy extracted from glucose so far. So all the energy is in those electrons. And so these molecules now are going to carry on their electrons. They're going to donate electrons to the electron transport chain, and then that's going to power the creation of ATP via oxidative phosphorylation. So all this happens in the mitochondria, and it actually happens in that little inner membrane inside of the mitochondria. So mitochondria are, they have two membranes. There's the outer membrane and then the inner membrane. And so that's where this happens. This whole electron transport chain is embedded in the membrane of that inner membrane. In eukaryotic cells. So in eukaryotic cells that have membrane-bound organelles, then these are going to be embedded in that inner mitochondrial membrane in eukaryotic cells. And then if you notice how the membrane is all foldy, that membrane is folded into these folds called cristae, and that increases the surface area. So like if you have more folds, you have more space to shove in more electron transport chains and make more energy. So having it folded increases the surface area for electron transport chains. There's not just one, there's a bunch in this membrane. And in prokaryotes, the electron transport chain is embedded just in the plasma membrane of the cell membrane, again, because they don't have membrane-bound organelles. So most of the molecules in the transport chain are proteins, and they usually exist in multi-protein complexes. So it's not just this protein, this protein, this protein. There's like complexes. So NADH and FADH2 will donate electrons to different electron acceptors early in the chain. And then the electrons are passed through several carrier molecules, including these molecules called cytochromes. So these are proteins that have heme groups containing an atom that is iron. And so these electron carriers and all these different steps alternate between reduced and oxidized as they accept and then donate electrons. So they're kind of playing like hot potato with all these electrons. And so the electrons will drop in free energy as they're transferred down the chain, eventually, finally passing to oxygen and forming water. And so the electron transport chain breaks this large free energy drop from glucose to oxygen into much smaller steps that release energy into manageable amounts. So remember in the intro part, I said it's kind of like that scene in The Martian where he's trying to make water and he's trying to do this reaction and it ends up in one big explosion. So this is why the electron transport chain is functioning the way it does, even though it's complex to learn about. It's so that it breaks up this large free energy drop into smaller steps and releasing energy in a little bit at a time and getting captured. And in the electron transport chain, all these electrons are getting transported, but no ATP is made directly by the chain. The ATP is made in the next part. So the electron transport chain happens to then drive the second part, which will make ATP. So this is just a figure kind of trying to show how that free energy change happens as electrons get passed along to different members of the electron transport chain. So this is kind of like a graph showing the free energy change. So as they move, it goes down a little bit, releasing energy in manageable amounts. So 2 electrons from NADH enter the chain, and then the electrons get transferred downhill through several complexes. So there's a complex 1, 2, 3, 4. Gets passed on, and then FADH2 passes on 2 electrons, and then they kind of, just play hot potato through these different protein complexes until eventually at the end, electrons get added to oxygen. It's the final electron acceptor, because remember, oxygen is very electronegative. It wants to take those electrons, and then we happen to make water. So this is kind of the summary of how it looks in the inner mitochondrial membrane. These are the different complexes, 1, 2, 3, and 4. There's a bunch of stuff there in the complexes that, we're not really like showing in detail. But the NADH feeds the electrons, and so this yellow arrow is showing the path of the electrons getting like passed along. So from this complex to this one, and then this one to here, cytochrome C, and then to this complex, and then eventually on to the oxygen to make H2O. So the yellow arrows are the path of the electrons. The black is just like the path of this molecule. NADH is now oxidized to NAD+. So that's that arrow. The red arrow is showing how when electrons get passed along, protons get passed along too. So remember I said that in the intro part. And so this is where that comes into play. So now as the electrons get passed on and they're bopping around between complexes, the protons are also getting pushed onto this side. So all the protons that are traveling with those electrons, so the H now, they end up on this side. So all of these protons are kind of just like building up over here. With each of those changes in electron carriers. So that's what the red arrow is showing, is like it's pushing the protons on that side. So the energy released as the electrons are passed down the electron transport chain is used to pump protons, or the hydrogen ions, from the mitochondrial matrix on the inside to the intermembrane space. So that's the space between the outer membrane and then the inner membrane. And so now the protons are like really concentrated on that side. And then the protons want to move down their concentration gradient back across the membrane because they all got shoved that way. So now the concentration gradient is really high out there and it wants to come back inside, right, from high to low. So when the protons are allowed to move down the concentration gradient back across the membrane, this is going to pass through an enzyme called ATP synthase. And what this does is makes ATP. So as the protons move into binding sites on the rotor of ATP synthase, it is like mechanical. There's like a part of it that's like a rotor, like a motor kind of. And so as the protons move into those sites, it causes it to spin. And then it spins in a way that catalyzes the phosphorylation of ADP to ATP. And so this is an example of a process called chemiosmosis. And chemiosmosis is the use of energy in a proton gradient to drive cellular work. So oxidative phosphorylation is one kind of chemiosmosis, and there is another kind of chemiosmosis that we'll talk about next lecture. So yeah, that's why it's those two parts, the electron transport chain and chemiosmosis, that make oxidative phosphorylation happen. So chemiosmosis is what couples the electron transport chain to actually making ATP. As these protons move down this ATP synthase molecule, protein, there's actually binding sites. So there's going to be another picture that's more detailed than this. But there's binding sites as it moves through on ATP synthase, and then it causes it to spin. This is like where the rotor is. And it spins in a way that makes a bunch of ATP as the protons go through. So it's kind of like a water wheel if you picture, like a river with like a cute water wheel and the water's moving the thing, and that might be generating electricity for something else. That's kind of what it is. So here you see a different figure of ATP synthase. That's a little bit more complex. Again, this is still super simplified, but here's those protons, the hydrogen ions. As they move through, they bind on one part, and then it kind of makes it spin. And then that spins this wheel, and then that's what's phosphorylating ATP. And then it spits out all the ATP inside the mitochondria in that space. That's the mitochondrial matrix. So, the combination of the electron transport chain and chemiosmosis makes the energy. The certain electron carriers in that electron transport chain accept and release protons along with the electrons, and maintaining that proton gradient is what couples the redox reactions of the electron transport chain to ATP synthesis. That H plus proton gradient is referred to as the proton motive force, emphasizing its capacity to do work. So protons, because it's protons, but then it has like a force to be able to move things and make work happen. So together, those make up oxidative phosphorylation. It's part one, the electron transport chain that makes that proton gradient happen. And then as they move down their concentration gradient, it drives chemiosmosis. And so together, that is oxidative phosphorylation. So that is cellular respiration. It's probably a lot, but hopefully broken up into manageable chunks. So harvesting energy from glucose has those 3 stages, just to summarize. Glycolysis breaks down glucose into 2 molecules of pyruvate. Pyruvate oxidation and the citric acid cycle complete the breakdown of glucose. So all those carbons end up leaving as carbon dioxide. But you're taking all the electrons and energy and putting them into oxidative phosphorylation. And then the electron transfer chain and chemiosmosis is what makes most of the cells ATP. So actually, here is a breakdown of how many are made. So in glycolysis, only 2 ATP are made. The citric acid cycle, 2 ATP are made. And then in oxidative phosphorylation, you make a bunch of ATP. So about 26 or 28 molecules of ATP. So that is what makes the mitochondria the powerhouse of the cell, is because this is where all the energy is made, and then especially oxidative phosphorylation. So in total, per glucose molecule, you make about 30 or 32 ATP molecules. So yeah, during the cellular respiration, most of the energy kind of flows in this sequence. It's kind of like passed on from glucose to NADH to the electron transport chain into that proton motive force, and then finally stored short-term in ATP. So about 34% of the energy in a glucose molecule is transferred to ATP, making about 32. And then the rest is lost as heat. So remember the law of, the second law of thermodynamics. Some is going to get lost into the environment as heat. But that's still pretty good. And so if you're like, wait, what do you mean either 26 or 28 ATP? Either 30 or 32 ATP? What? So there are 3 reasons why the exact number of ATP produced is not known. Because 1, photophosphorylation and the redox reactions aren't directly coupled. So that ratio of NADH to ATP molecules is not a whole number. 2, ATP yield varies depending on if the electrons are passed NAD+ or FAD. And then 3, the proton motive force can also be used to drive other kinds of work. So this is kind of the best estimate that we have. and that's it. Okay, so that is cellular respiration. And then up next is fermentation.